What is the basic characteristic diagram? Gram-positive, human normal flora, dislocation infection, pyogenic abscess such as fevers, pneumonias, sore throat and ear infections. Then classification of streptococci. So these are broadly categorized into two types. Streptococci are broadly categorized into two types. Aerobic streptococci, anaerobic. Okay, both are aerobic only. Very good. That means these are strictly aerobic bacteria. They need oxygen. They are not anaerobes. Okay. Then these are further divided into facultative when obligate. I hope you understood what is facultative and obligate. Facultative means they can survive even in the absence of oxygen. They can survive even in absence of oxygen, but they cannot survive in the absence of oxygen. They always need oxygen. Obligate oxygen dependent and facultative aerobics. Both are aerobics. There is no anaerobes. Okay. So obligate aerobics, Streptococcus. This is a very rare bacterial infection. This is example PEPTO. Streptococcus. Streptococcus is the example for obligate aerobic streptococcus. Then and facultative aerobic streptococci. These are further classified based on hemolysis on blood agar. Based on hemolysis on blood agar, these facultative aerobic streptococci are divided into three types. Okay. So can anyone say what type of hemolysis we will see in blood agar? Do you remember? There are three types of hemolysis: partial, complete, yeah, and other one, discoloration something I forgot. Yeah, they are called as alpha hemolysis, beta hemolysis, and gamma hemolysis. Very good. Gamma hemolysis. Based on the hemolysis, these are further classified. Clear? Let us see. So anti-streptolysin O. Okay. Anyway, we'll see. So alpha hemolysis is seen in Streptococcus pneumonia. Example: alpha hemolysis is exhibited by Streptococcus pneumonia. Beta hemolysis. Oh, now we got another problem. Okay, beta hemolysis is shown by many streptococci. How to classify them again? Beta hemolysis will be shown by almost all types of Streptococcus species. That means just this is not sufficient. Got my point? Most of the Streptococci will show beta, but beta has many species, and I need to pinpoint the type of bacteria. Got my point? How we will do? Then we are going to take another approach. When we get a beta hemolysis, then this beta hemolysis samples will be subjected to lancified serotyping. There is something called lancified group. Lancified serogrouping. So what we will do is, we will take a sheep serum and we will add to this one, to the colony. Okay. So lancified serotyping. Based on lancified serotyping, these are divided into, write down, lancified serotyping A, B, C, D, and there are up to E to H also available, by the way. E to H also available. So I am not sure what is exactly lancified serotyping done. Maybe you can check in the textbooks later on and online. So based on this lancified serogrouping, we are categorized them A to H. Okay. If we get a positive, lancified grouping A positive, then it might be as pyogens. Pyogens will have lancified A positive. Lancified B positive will be aeruginosa. Streptococcus pyogens, Streptococcus aeruginosa. Then C positive will be seen in Dysgalatia, Streptococcus Dysgalatia. This will cause a C positive. Then D positive is seen in Enterococcus. Enterococcus fecalis. Enterococcus fecalis. And I came to know just now, after a long time I am saying microbiology again. E2H, lancified E2H are animal infectious strepto, not in humans. E2H will be infecting the animals, not humans. E2H positives are animal streptococcal infections, not the human streptococcus infections. Okay? Very good. So now, in today's lecture, we will study about a few important streptococcus species. We will study about the streptococcal pneumonia. What is this infection? What is this characterizations? We will study about streptococcal pyogens, that is pus forming, and fecalis. Can anyone say fecalis means it will cause some entero infections, intestinal infections, pus forming infections, lung infections. You understand it, right? No? And aeruginosa is skin infection. Skin, pus, respiratory, gastrointestinal. Okay? So now we will focus on S pyogens. So we will focus on Streptococcus pyogens. What is this Streptococcus pyogens? Okay, Streptococcus pyogens. Okay, write down. Write down the properties of Streptococcus pyogens. Properties of Streptococcus pyogens. It will cause pus forming skin abscess. It will cause pus forming skin abscess on the body. It will cause pus forming skin abscess. Pus forming skin abscess. Properties: size 0.5 to 10 micrometers in diameter. Properties: size 0.5 to 10 1.0 micrometers in diameter. Shape: round. It is round in shape. Color: it is colorless, and beta hemolysis will be shown. See, now we are connecting it. It will show beta hemolysis. It is round. It is round shape, colorless, beta hemolysis. Next one, gram positive. It is gram positive. Non-motile, and capsule is present. Polysaccharide capsule is present to this one. Okay. So, S pyogens groups. There are two types of S pyogens. By the way, students, I understood. These bacteria classified based on the diseases. S pyogens is a skin disease. It's a condition. Streptococcus mediated pyogenic infection. So this skin pus infection can be shown by S pyogens and S aeruginosa both. So yes, we are studying about pyogens anyway. Okay. So classified grouping will be done on. These are classified under classified group A and B. S pyogens is in group A. S pyogens belongs to group A. Classified group A. S pyogens belongs to classified group A. Yeah. I need to erase the board now. Culture properties of S pyogens. What cultures you will take right now? Oxygen requirement. Oxygen is mandatory, or even it can also grow at 10% CO2 in the medium. Okay, oxygen requirement. Write down in culture requirements you are writing. Oxygen 10% CO2 is required to the medium. Why it means 10% CO2 in the medium? Because this is a facultative. That means it can even survive in absence of oxygen. So we are mimicking the conditions. To the medium we are adding 10% CO2. Temperature 37 degrees centigrade. Temperature 37 degrees centigrade. Types of media. We can use blood agar media, sugar media and sugar media. The purpose of sugar media is fermentation of carbohydrates. Carbohydrate fermentation. Sugar media. Medium include blood media and sugar media. The purpose of sugar media is for fermentation studies. Fermentation studies. Because they are classified under fermentation studies also. That's it. Okay. Incubation period. Incubation period. Incubation is around 24 hours. Incubation period is 24 hours. And colony morphology. Upon the growth of colony looks like circular cormets. Circular colonies or convex colonies can be seen. Circular or convex colonies can be seen. Okay? Alright. Next one. Biochemical characteristics. Biochemical characteristics of aspiogens. Biochemical characteristics. Biochemical characteristics of aspiogens. They are catalase negative. Catalase negative. Next one. On 10% bile, they are non-soluble. On 10% bile solution. 10% bile solution, they are non-soluble. They won't soluble in 10% bile solution. Sensitive to penicillin, they are penicillin sensitive. That means they will be killed by penicillin easily. Penicillin sensitive. Vogus crossover test. They are negative in Vogus crossover test negative. Vogus crossover test negative. Vogus crossover test negative. Gas production nil. They won't produce any gas. How we will check whether they are producing gas or not? By sugar fermentation test. Right? Gas production negative. Gas production negative. Yeah. So these are, now you know the properties, you know the culture properties, then you know the biochemical properties of aspiogens. Am I correct? Yeah. Then next one. Next one write down. Group A aspiogens. Group A aspiogens. Group A means lancified group A aspiogens. Right on. They will cause two types of infections. Pyogenic infections and non-pyogenic infections. This will cause two types of infections. Pyogenic infections and non-pyogenic infections. Pyogenic infections and non-pyogenic infections. Right on. Now, first of all, pyogenic infections. Pyogenic infections include respiratory infection, scarlet fever, skin infection, deep skin infection, not skin. These are the four infections caused by pyogenic. Respiratory, scarlet fever, skin and deep infection. Right on in respiratory, tonsils. Now you understand, right? What are tonsils? Tonsils are just the streptococcal infection to the throat. That's why when you have this throat infection, there will be amoxicillin, which is an antibiotic. Okay, yeah. Tonsils in respiratory, sore throat and tonsils. Then skin infection include erythematous rashes can be seen on skin. Erythematous rashes, red rashes can be seen on the skin. Then Scarlet fever you already know, it's just a fever, okay? Then deep infection. Sometimes this strepto will cause deep lung infection. They will cause lung infections. They will cause brain infection, kidney infection, and even liver infection can be seen in severe pyogenic infection. Lung, brain, skin, liver and kidney infection can be seen by as pyogenes. See, a skin normal flora causing deadly consequences in the body. It is present as a simple skin bacteria, but it can kill your brain. It can affect your kidney once it is dislocated. Okay? Yeah, very deadly. Then, non-pyogenic infections. What are pyogenic infections we will see? Right on. Glomerular nephritis. Glomerular nephritis, that means kidney infection, okay? Glomerular nephritis. Glomerular nephritis. And other one, rheumatoid fever. Rheumatoid fever. Rheumatoid fever happens because of inflammation to the lungs. Lung inflammation will cause elevated body temperature. Lung inflammation will cause elevated body temperature. That is called as rheumatoid fever. Is it clear? So now we understood the clinical presentations of Group A. Okay? Right or not? Group B, parallel you just write here itself. Group B will cause meningitis, pneumonia, neonatal sepsis. Group B will cause meningitis, very dangerous, pneumonia, neonatal sepsis. Meningitis means brain infection, skin infection, pneumonia and neonatal sepsis. Just born babies will have this infection. Now I will tell one interesting thing. These are caused by Streptococcus galacticea, A galacticea. I don't already here, right? Listen, Streptococcus A galacticea is the skin normal. It is the normal flora of female's vaginal cavity. This is the normal flora of female's vaginal cavity. Listen, once they are undergoing the delivery, once the delivery happens, the baby comes through the vaginal cavity and the baby will expose to the A galacticea. And A galacticea is not normal flora to the skin. So baby will catch a neonatal sepsis. Okay? I want to show the pictures of this, then you will understand. This will cause napkin dermatitis. Just born baby within 3 to 4 days, just like the snake will remove its skin, right? The baby's skin will start shedding off with severe infection. Baby will cause, severe discomfort to the just born baby. And babies won't have much immunity also. These complications can be seen in Group B, Streptococcus, that is Streptococcus A galacticea. Whereas pyogenes will cause the things we discussed. Then Group C, what they will cause? Group C will cause pharyngitis and endocarditis. My God, endocarditis is that is the heart infection. See, heart is always under movement. Even the moving heart is affected by bacteria. How strong it is? It is, yes. Streptococcus dysgalacticea will cause endocarditis. Streptococcus galacticea will cause endocarditis. Okay. Then Group D. Group D will cause GI infections. You already understood. Group D itself are further classified into two types: Enterococcus and non-Enterococcus. Group D are two types: intestinal and non-intestinal. Intestinal is Enterococcus fecalis. This will cause urinary tract infection and wounds can be seen. So, if a patient has urinary tract infection, possibility is there that it might be a Streptococcus Group D, Enterococcus fecalis. Enterococcus fecalis can cause UIT, urinary tract infection. Whereas this one, non-Enterococcus is seen in animals again, bovine infections. Cattle will have, cow will have some infections, okay? Bovine infection C. Alright. Oh, I successfully completed almost this bacteria. Okay, good. Now write down next one. One important thing I need to say. Antibiotics. Keep aside writing antibiotics. See, each and every bacteria, naturally they are resistant to some antibiotics and are sensitive to some antibiotics. Each and every bacteria, normally. Okay? So our pathogens bacteria are sensitive. Sensitive means they will die. They are sensitive to write down. Bacteriacin sensitive. They are bacteriacin sensitive, penicillin sensitive. They are bacteriacin sensitive and penicillin sensitive. That means we can kill them by bacteriacin and penicillin. Okay? Bacteriacin and penicillin. Then resistance. They are resistant to sulfamethoxazole. They are resistant to sulfamethoxazole. Sulfamethoxazole. They are resistant to sulfamethoxazole. And I just want to tell one more thing, students. Listen, this bacteria has a surface antigen. All the Streptococcus bacteria has, pathogens has a surface antigen called streptolysin. Streptolysin. Okay. So we will go, this is an antigen, and the streptolysin antigen can be detected by using antibodies that are active to the streptolysin. These antibodies are called as anti-streptolysin, ASO, anti-streptolysin O antigen. This is O streptolysin antigen. That we will see. Keep these properties. Then moving forward, lab diagnosis. Lab diagnosis. Write down everyone, lab diagnosis. So we are going to understand the diagnosis, okay? The diagnosis approach of this Streptococcus pyogenes. So first one, specimen. What specimen you will take for Streptococcus pyogenes? We already understood its behavior, right? Now tell me, what will be the ideal specimen for diagnosing this pyogenes? Skin sample, stool sample, urine sample, abscess, blood sample. Why you will take blood sample? Actually, it won't present in the blood. Right now, throat swab, wound swabs, blood. Yeah, blood came. Maybe due to systemic infection, deep infection, brain infection. Blood sample might be taken. Correct. Blood sample, pus sample, UIT, urinary tract urine sample simply. Urine and genital swabs will be taken. These are the specimens. Then next one, collection and transport. Next slide please. Yes, mister. Yes. So collection and transport. Collection and transport. Specimen collection and transport actually. Right now, it should be taken in a sterile container. And it should be transported in a container. It should be transported in a container that having 10,000 crystal violet sodium azide. 10,000 crystal violet. Okay, simply write down that's having crystal violet sodium azide. Crystal violet plus sodium azide. That means sodium azide and crystal violet will act as a preservatives during transport. Azide, A-Z-I-D-E, okay? Sodium azide are the preservatives, okay? Then culture. What culture you will do? Blood culture. Write down first one. Cultures, blood culture. At 37 degrees centigrade for 24 hours. Incubation time 24 hours. Morphology. Asparagines morphology, write down. Circular, low convex, colorless, beta hemolysis, opaque colonies will be seen. Circular, low convex, beta hemolysis, opaque colonies will be seen. Opaque colonies will be seen. Opaque colonies will be seen. Then biochemical tests. Biochemical tests. Yeah. Write down in biochemical test. Catalase negative. 10% bile non-soluble. PVR negative. Vogus crossover test. Biochemical test. Catalase negative. 10% bile non-soluble. Listen. In 10% bile non-soluble, within the bracket, write down soluble for Streptococcus aureus. 10% bile soluble is for Streptococcus aureus. Where is Streptococcus aureus? Where we wrote Streptococcus aureus in our classification? Pyogenes, Aeruginosa. Yeah. Aureus is also, Aeruginosa and aureus are normal flower, okay? So how to differentiate aureus from pyogenes means? Pyogenes is non-soluble whereas aureus is soluble, okay? Yeah. Lancified serotyping. Yes, students.
So this is an announcement regarding CUFest 2024. There will be inter-department and inter-department competitions. There are 6 domains: dance, music, modeling, literary, finance and theatre. Okay. If you are interested, you can give your names to your event coordinator. You know who your event coordinator is. Okay. So you can give your names to her. And the winners of the inter-department competition will be sent for inter-department competition, that is on the CUFest days itself. And the final winners will be awarded with cash prizes and scholarships and training according to your capability and interest. Okay? And be sure that you give your names as fast as you can, because after 3 to 4 days, the inter-department competitions will start. Okay? Thank you, sir.
Thank you, sir. Yes, students. Awesome. Lancified grouping. Lancified grouping. Yeah. What will be the lancified grouping here? What positive we will get? Yes. Lancified grouping A positive we will get for S pyogenes, because that is what we see in classification. Okay, S pyogenes is lancified grouping A positive. Right now. Lancified grouping A positive will be seen. In case of rheumatoid, next point. In case of non-pus infections, we will go with anti-streptolysin O test. Anti-streptolysin O will be positive. Anti-streptolysin O positive for S. aeruginosa. Anti-streptolysin O positive will come for S. aeruginosa. Okay? Then treatment. Last of all, treatment. Penicillin G is the drug of choice. Penicillin G is the drug of choice. Penicillin G is the drug of choice. Prophylaxis. Last of all, prophylaxis. What is the meaning of prophylaxis? Prevention. Okay, prophylaxis. Yeah. So what preventive measures we can take? So penicillin should be administered in children who is having signs and symptoms of sore throat. Okay. In children, whenever they are having signs and symptoms of sore throat, penicillin can be given because this is a penicillin sensitive. Alright. That's it. That is all about biologics.